So last time we left off talking about inhibitory synapses, and just to recap, we divided synapses to be either excitatory or inhibitory, and we said excitatory synapses usually involved the opening of sodium and calcium channels, right, which allowed those positively charged ions to enter the cell, and that would depolarize the cell and lead to an action potential. Inhibitory synapses usually involve the opening of potassium and chloride channels. Okay. And let's take potassium as an example first. When we open up potassium channels, potassium will move out of the cell, and this will eventually drop or decrease the membrane potential to become more negative, and this will hyperpolarize the cell, making it harder for an action potential to be fired here. Okay? So as a result, this is an inhibitory postsynaptic potential, opening of potassium channels. The other type of inhibitory synapse is involving chloride channels. Now, chloride channels have two different types of functions in terms of inhibiting or having an inhibitory response at the synapse. So if we opened up chloride channels, this would result in the movement of chloride into the cell, and chloride is a negatively charged ion, and so that would have the same effect of opening potassium channels, which is to decrease the membrane potential, make it more negative, or hyperpolarize the cell. That, in effect, will make it more difficult to fire an action potential. Now, this is specifically when we have an active transport of chloride across the membrane. So we know that chloride channels have two different functions. We can influence chloride to move into the cell if there's actively chloride being pumped out of the cell. So I'll say that again. If we have active transport of chloride, we create a gradient where chloride will then move back into the cell. That will be an inhibitory postsynaptic potential. The second way that chloride can influence the membrane potential is to stabilize it. If we have more than one type of signal, opening up a chloride channel will stabilize the membrane potential. So let's talk about what we mean on this slide. Okay, so here, what we see in the image on the left is that both, there are no active transport of chloride, there's no active channels of chloride open, and so chloride is moving freely and it's at equilibrium here. Okay, there's no active transport. Now, if we had a different type of signal, if we had an excitatory synapse here, so neurotransmitter 2 is an excitatory synapse, neurotransmitter 1 is an inhibitory synapse. Chloride in this image, since there's no active transport here, will basically negate that excitatory stimulus. So I'll say that again. This is a neuron where we have two different synapses. We have one, which is inhibitory from chloride. We have two, which is excitatory. So we're opening up a sodium channel here. We're bringing sodium into the cell, but we're also bringing chloride into the cell. But chloride is a negatively charged ion. It's going to negate or cancel out the positive that we're bringing into the cell. So in this way, the opening up of chloride channels stabilizes the membrane potential. We prevent depolarization, cancel out the effect of having that sodium channel open. All right, so not only do chloride channels hyperpolarize the membrane, but they can sort of cancel out or negate any depolarizing stimuli and basically stabilize the membrane potential. Any questions on that? Okay, so let's jump back on this slide here and define or describe these inhibitory postsynaptic potentials. So first of all, we want to remember that these are graded potentials. We spoke about graded potentials earlier, and these are the types of graded potentials that we were referring to. Okay, so the example here is that if we have a higher frequency of an action potential And a higher frequency just means we are firing more action potential, like firing at more frequency. What we induce is more neurotransmitter being secreted at the axon terminal. So a higher frequency translates to more neurotransmitter being released. That neurotransmitter will then cross the synaptic cleft, bind to receptors on the postsynaptic neuron, and either open or close those inhibitory channels. Okay. Let's take potassium in this example. So let's say we had a higher frequency of action potentials in our presynaptic neuron. This stimulated more neurotransmitter release. This neurotransmitter binds to, let's say it's a potassium channel on the postsynaptic side. Okay, binding to that potassium channel will open up those channels and we'll have potassium being moved out of the cell. Okay. So this basically increases the ion permeability across the membrane. We increase the movement of potassium out of the cell. Okay. And this basically leads to greater hyperpolarization. So all this means is that increasing the frequency of the action potential basically increases the magnitude of the stimulus. If we have action potentials being fired at a faster rate, more frequently, we will trigger a bigger or greater hyperpolarization on the postsynaptic side. Okay, and that's what we mean by graded. We can have a small stimulus, we can have a medium stimulus, we can have a large stimulus. Can we have that with action potentials? No, action potentials are not graded. Okay, you cannot have a small versus medium versus large. It's all or nothing. Inhibitory postsynaptic potentials are graded. They are graded. Okay, and the way we can grade them is by influencing the frequency of action potentials on the presynaptic side.